right. Good morning, everyone. Welcome to Population Genetics 3. My name is JP, and I'm going to be giving the first talk of this session. I believe the gong is the start for us. So thank you very much. I know it's a little bit early. I'm going to be talking a little bit about this research that I'm developing with Patrick James at the University of Toronto. We're interested in looking at the variability of outlier detection methods in a very interesting system that involves like population outbreaks. I always like to start these kinds of talks reminding ourselves that landscapes are usually not even. We have a lot of heterogeneity going on. It's composed of different habitats, different environments, different barriers that act differently, and I'm sorry I'm using a lot of difference in the same sentence, that acts differently for the diversity that occurs in that landscape. So that also reflects on the distribution of diversity across that landscape. When you're talking about these diversity patterns, you also have to remember that we have these interactions between environment and genotype, and that translates into phenotypes, and that will eventually impacting on how that distribution is, how this diversity is distributed in that landscape. So looking at these spatial patterns of genetic diversity in this landscape, this heterogeneous landscape, can provide us insights of the evolutionary processes, the evolutionary forces, the selection pressures that are behind this generation and what's maintaining it. These genetic diversity patterns, we can be very simple here and remember there are two main evolutionary drivers behind it. We have adaptive variation that relate to selection, selective pressures, but we also have neutral variation that it's non-directional, and I'm going to talk a little bit about that. This neutral evolution, we assume that there is no directionality. We don't have a selective pressure that is biasing the frequency towards something. Those changes in frequency, those changes in composition happens through random sorting of the individuals and genomes through time and through space, with uneven sorting of that diversity as the population develops. This kind of evolution is useful for us to discuss patterns of connectivity, gene flow, differentiation and structure, and demographic variation across an area. But when we're looking at adaptive evolution, we're thinking about that directionality. We're thinking about how these genetic variants are favored over a selection gradient. So what's happening in that environment that will end up impacting the genetic composition that we have? And kind of like think as an evolutionary response to selection. So what is being selected, what's changing, and why that is happening? One way to explore that is by looking at outlier loci. Outlier loci would be regions of the genome genes that present a higher frequency than what is expected by neutral patterns alone across a gradient. So if you can look here on the top right, we have these red and blue lines where you have changes in allele frequencies in response to environmental variable, as you would not expect to see that in that black line with neutral selection. Neutral evolution. One way that we can find these regions is by applying genomic scan methodologies, and we can be very simple about it. We have two main ways to approach this. We have differentiation methods, where we have comparisons of frequency between and within regions of the genome, and you would flag regions and outlier loci that have a significant deviance of what is expected from a normal distribution, neutral distribution, sorry. And we also have the genotype environment associations that would flag those outlier loci based on correlations of allele frequencies and those explanatory variables based on our environmental gradient. And then it's great because you can start using that adaptive variation, that local adaptation signals to infer of what's driving evolution between environments or around barriers or across distributions. Which is great if you have a genome, you can map that to the genome and see which genes are being involved, what signals of selection we have. You can talk about adaptive potential, resilience, but most of this analysis, they assume some sort of stability through time and space. It's either static or you have a predictive linear growth, which is not what happens in some systems. For example, we have systems that vary their densities and distribution really fast in this explosive manner that what we call like a population outbreak. So very simplistic, we imagine that this would be a regular growth, put that on steroids, you have a lot of individuals happening from generation to generation across the distribution, and that's what we would expect on a population outbreak. We see this behavior across the taxonomic tree. We have mammals, we have fishes, we have insects. We have similar behaviors in invasive species where that expansion ends up taking over the landscape. And depending on what we're talking about, for example, with forest defoliators, that's the example that we're going to be using here, that accounts for a lot of socioeconomic importance, industry, so you do have a lot of interest in ecology, in conservation, in management, in systems that behave like that. At the genetic level, what is happening? Once you have this explosive expansion, imagine that you have a series of leading edge effects. So we have allele surfing, we have bottlenecks and founder effects happening unevenly across that expansion. So you have more individuals at the edge of the expansion interacting with each other, which end up creating differences between areas of that distribution and through time. And to make it a little bit more interesting, you have some species that present this in cyclical patterns, where you have expansions and contractions from endemic phases where we have less connectivity, more isolation, to an epidemic phase where you have basically a metapopulation that interacts randomly across their distribution. And that brings that attention to the ecological context of the time of sampling. The timing of sampling matters in these systems because you can see here, for example, in the literature, same organism, same area, sampled at different times. You have different results about connectivity, different results about diversity. You see that changing through time. And particularly in these forest defoliators, you also see that changes through space. So depending on where you are, when you are, you can get different metrics of structure, connectivity, diversity at the same time, and it varies from 1 year to year. So we know that the ecological context, the time of sampling is important for these neutral variations, but what is happening at the adaptive level? How is that outlier inference affected by those changes in demography through time? So Patrick and I decided to investigate the behavior of some of these methodologies, what's happening with that outlier detection through time in an outbreaking system, and how does that relates to different ways of exploring outlier loci. Our study system, we went for one of those forest defoliators here for North America, from the boreal forest, the eastern spruce woodworm. If you're from around here, you probably read or heard about this. It's a very big forestry industry here. We took some samples that some individuals that were sampling through the current outbreak that started developing in 2006. We are not here in Quebec. That right now is not an area of edge expansion anymore. We're close to the epicenter. We're using 4 different sampling years, 2013, 14, 15, and 17. And these data sets have been studied for that neutral variation, and we see that matters. So the timing here, we're dealing with a data set that we know that differences in neutral variety show up when we do our analysis. So we have a SNP data set. We sequence with genotype by sequencing, sampled close and far from the epicenter in these different years, and we are analyzing each year separately. So imagine that you have one sampling season, what would you see? Another sampling season, what would we see? Cause of distribution. For the outlier detection methods that we're going to be using, we're going to try to use, we went to the literature to see what's more common, what you see a lot. So for the differentiation methods, we're going, differentiation-based methods, we're going for outflanks. And then from those GEAs, we have an univariate approach, which is our LFMM, and a multivariate, which is our redundancy analysis, all performed in R. And for our explanatory variables, we went to the literature, we saw which one of those climatic variables would be important for the development of the outbreak in this species. and we filtered some of them to see if there's any adaptive correlation there. So what do we see? Well, first, we see that the number of outlier loci detected varies per year and varies per method. But the trend is kind of similar. We have a higher number in the earlier samples, lower number in the lower samples. But we do see here this peak in 2014, which coincidentally is our bigger year for sampling, bigger data set. So what we decided to explore, like what would be the effects of that size. So we subsample those first 2 years that are bigger data sets into those smaller data sets that match the last year, so 145. We did that randomly a few times, kind of like in a bootstrap approach, so we do have like these distributions of values now. And depending on how we subset that data set, we have different results, and for all methodologies, and it varies from year to year. So bigger data sets, you have more outlier loci, which is kind of what we'd expected. If you have a bigger, there's a higher probability of real sampling of the diversity in bigger data sets. But depending on how we do the subsets and what part of the diversity composes that subset, our outlier flagging changes. Within the same year, comparing those methods, we see that the outlier ID and number vary. So what we're seeing here, some Venn diagrams, I don't think it's very good here on this on the projection, I'm sorry, but each one of them is a year. Each one of the smaller circles is one of those methodologies, and we want to see if there's any overlap between the outliers. And our differentiation method, outflanks, have higher number of candidate loci, mostly unique to that methodology. Higher congruence happens early in those earlier years with bigger data sets, and the farther we get, like the later years, those loci are unique to each method and they don't overlap. Now, if you look within the same method, so we're looking, each one of those graphs is the outflank for every year, LFMM for every year, and RDA for the 2 years, because RDA didn't detect any outlier loci in 2015 and 2017. We see overlap in the earlier years, bigger data sets, most unique outlier loci in these bigger data sets as well, and a higher number in our differentiation method, which we think that might be related to artificially inflated frequencies due to those bottlenecks and allele surfing, where we have higher frequencies of neutral alleles than expected because of the uneven sorting of the population as it expands. So some take-homes here. Time of sampling in outbreaking system is important for outlier delineation, as well as the sample size and the sample composition. Things change really fast throughout the area, throughout the distribution. So that matters when we're planning on how we're going to be approaching this with a GA methodology. It's a similar in-between quotation mark problem that you see with the neutral variation, so the timing also matters here and the composition. The outlier detection doesn't seem to be intrinsic to the system, but it seems to be intrinsic to the sampling, on how you design your sampling and how your samples is composed in time and in space. So considering that, considering sampling composition, considering the time of sampling in the ecological context, is very important to avoid confounded conclusions in eco-evolutionary inferences of local adaptation and any implications that might take. So with that, I'd like to thank a bunch of people that has helped us through the years. I love the funding agencies. I'll take some questions if I have time. You can reach me out an email as well, or later on in the conference. Thank you very much. It just like the frequency of that same loci wasn't high enough for those methods to apply to flag it. So if the composition of data set doesn't sample properly the population for the variant, I think because on how those outliers are determined, it was just not flagged out. So it was kind of unexpected, but cool at the same time. So yeah, thank you very much.
Okay. All right. Hello, everyone. So changing tactics a little bit now to DNA methylation and epigenetics. So my name's Hollie. I'm a new group leader at the University of Leicester in the UK. And today I'm going to talk to you about epigenetic change across evolutionary timescales and how I think this plays a role in genomic adaptation. So within my lab, we focus specifically on DNA methylation, which is one type of epigenetic mechanism. And we look at this in arthropod species. So for those of you who aren't familiar with DNA methylation at all, or maybe you know it in other contexts, I'll explain it a little bit. So in our arthropods, we primarily have gene body DNA methylation. So this is where it's found within the exons and introns of a gene. Generally speaking, we don't have promoter methylation in these species, although there is the odd one that does have it. And it's a mixed bag whether or not we have TE methylation. Some species do, some species don't. Within our arthropods, it's in very low levels. Again, if you're familiar with things like vertebrates, it's much lower in the arthropods. And generally, it's found in this CG context, so a C followed by a G. Within arthropods, it's associated with higher, more stable gene expression. So again, if you're familiar with promoter methylation in other species, this is almost the opposite. So it's associated with higher levels of gene expression. But across the arthropod literature, generally speaking, if whatever study, whatever they're looking at, if they found differential DNA methylation, it's usually not correlated or it doesn't correspond to differential gene expression within these species. And so this really leaves the question of, well, what is it doing? Why is it there? And so I have a bit of an idea as to what I think it might be doing, and this idea actually is cropping up in the literature in various species now, which is really exciting. If you were in the symposium yesterday, this came up. So we know that DNA methylation is a mutagen. We know that it mutates cytosines to thiamines at a rate much greater than the background genome mutation rate. And we also know in our arthropods, as I mentioned, it's found in gene bodies, and usually in exons, sometimes introns, but usually exons in most of these species. So what do we have in exons? We have codons, which give us amino acids, right? Now, we know that the codon code is degenerate, so a certain substitution at a given base may or may not change the amino acid that it codes for. So I think most people here probably know this, but a very quick tour of what that means. So if you've got a nucleotide that's a zero-fold degenerate site, that means every time you get a substitution at that nucleotide position within the codon, you will get a different amino acid every single time. If you've got a site that's a four-fold degenerate site, that means any time you get a mutation at that site, there's no amino acid change. And so the idea here is if you have a mutation at a zero-fold site, new amino acid means potentially new protein doing something slightly different and potentially phenotypic changes, substrate for selection. And so this got me thinking, what if DNA methylation is enriched at either end of this spectrum? That could tell us something about what it's doing. So if it's enriched at zero-fold sites, we might suggest that maybe it's there as a mutagen in order to provide genetic change and thus substrate for selection. But if it's enriched at these four-fold sites, this might suggest that actually the genome has evolved to protect itself from this mutagenic quality of DNA methylation. So when I was thinking about this, at the time I was sat on a load of bumblebee genomes and epigenomes, and so I had a look in my bumblebees, and funnily enough, found that DNA methylation was enriched significantly at zero-fold degenerate sites. So this is the proportion here. So this is only Cs, and only in a CG context. And so it's enriched at these zero-fold degenerate sites where when we get a mutation, we will get a different amino acid, potentially substrate for selection, right? And so I thought, okay, maybe this is one genome, it's one species, or well, a few genomes, but one species. And so at the same time, I was also working on the Daphnia magna genome, so that's a water flea. This is a bar plot because it's only one genome, not multiple ones. And again, we've got enrichment at zero-fold and two-fold degenerate sites of DNA methylation, and we know it's a mutagen. And as I just said, if you get a mutation at these sites, you're going to get a different amino acid. So this is really now what my lab is trying to pursue. It's trying to figure out, is this actually doing anything? Is this actually contributing to genetic diversity within populations? And so that's what we're looking at. So to try and get at this, I've been collaborating with the University of Birmingham, so Professor Louisa Orsini's group. Louisa is here, so if you're interested in this system and you're interested in the genetics of the system, go find Louisa. But if you're interested in the epigenetics, then you can come talk to me. So let me explain our system. So we work on water fleas, and they reproduce by cyclic parthenogenesis. So usually they reproduce clonally, so you have genetically identical female offspring to the mother. And that's really important for our epigenetic studies because, as you will have heard a lot yesterday if you're in some of the symposiums for epigenetics, the genome can really dictate what the DNA methylation profile of an individual looks like. So here we can control for that because we've got this nice clonal reproduction. But they also reproduce sexually. And so when they produce sexual eggs, these eggs are in a seed-like pouch. And what will happen is the eggs will float down into the sediment of a lake and remain dormant until conditions become favourable to hatch. Now, not all of these eggs will hatch straight away because maybe more sediment will fall on top of them and they don't get to see the nice favourable conditions. And so as scientists, we can go along, take a sediment core of a lake, carbon date the different layers, fish out these little resting stages, and hatch them in the lab. So we can actually study populations from hundreds of years ago, living individuals of them in our lab, which is really cool, right? I think it's cool. And as a quick aside, the oldest ever resurrected Daphnia was 700 years old. So 700 years ago, it was created sexually, lay dormant, and someone's hatched it 700 years later. I think that's pretty cool. So in our study, we don't have 700 years, but we do have back to 130 years. So Louisa went off to Denmark, took a nice sediment core of this lake with a really well-characterized environmental history, and then hatched a bunch of these Daphnia from various time periods. So we have these temporal populations. So we know the lake was in pristine conditions up until about the 1930s, and then it underwent this really extreme eutrophication event due to sewage inflow from a nearby town. That then stopped. The town fixed the problem. So we have this really distinct time period of eutrophication. We then have a later distinct time period of pesticides, agricultural runoff into the lake, and again we see that in the chemical data of the sediment. And so we end up with these 4 temporal populations from the same spatial location, separated through time, hatched in the lab, and we have nice genetic lines of all these populations. So what did we do with this? So we press the button. So we threw a load of sequencing at it, basically, at all these different genetic lines to try and figure out what is going on across this evolutionary time period in response to all of these environmental changes. So the first thing to show you, it's not the best graph, but I'll explain, is that we looked at open and closed chromatin using ATAC-seq across all of these different genetic lines across this time period. And again, to be honest, I expected to see nothing here. I thought maybe it was a bit of a waste of money initially. But really surprisingly, we see a great big opening of the chromatin straight after this pristine population. So again, this is population data. So we've got 9 to 12 genetic lines for each of these phases. And then after this pristine phase, the chromatin opens up, and it's mostly on one particular chromosome in one region. So all of those blue dots are showing you regions of open chromatin, and it all seems to be localized to chromosome 10. So, of course, the big question, what's in that region? I see a few people smiling. Yeah, we don't know. So there are a bunch of genes in there, but they're all unannotated because it's a non-model organism, and they're all uncharacterized. But we do have a new genome now, so ask me again in 6 months, and maybe I can answer. But the point is, we have this population response in chromatin over time here. So then we looked at the DNA methylation, and we don't see any temporal population clustering of the DNA methylation profile between these different chunks. And so the majority of the variation within our DNA methylation is very much captured by the genetics of the individual. So each genetic line has its own unique DNA methylation profile. And so we've done lots of experiments in the lab on these lines, and we have a few bisulfite sequencing data sets for the same line at different clonal generations in controlled conditions, just by chance, because we were doing other experiments. And it's really highly conserved. So this methylation does not change in our lab over clonal generations. It's very much linked to the genetics. So we also looked at microsats a while ago for the genetic diversity in this population. This is Louisa's work. We don't see any population structure by microsats, but we do have whole genome sequencing en route. And so, again, you can maybe ask Louisa about the sequencing of the genomes part. And then the last bit, and I think this, for me, this is the most interesting bit actually so far. So because we have these genetic lines in the lab, we can do experimental work on them as well. And so what I did a few years ago was to take a bunch of the genetic lines that had historical pesticide exposure, so the ones from this pesticide time period, take a bunch from the pristine period that have never experienced pesticides, and expose them to pesticides in the lab and say, okay, what happens here? Is one fitter than the other? We did some DNA methylation sequencing on this, and again, this was really surprising to me at the time. We actually find that the population whose clonal ancestors had experienced pesticides in the wild, whenever it was, in the 1980s, these ones show a massive change in DNA methylation all across their genome when you expose them to pesticides again. And this is both hyper and hypomethylation, so changes all over the place. But the population that never experienced pesticides, they don't change their methylation, hardly anything, just a few changes. And so this was, really surprising. And so I have some theories on why this is happening, and maybe we can, if you're really interested, we can talk about this afterwards. Okay, so to summarize what I've just gone through with you. So I've told you that DNA methylation in bees and in Daphnia is enriched at these 0-fold and 2-fold degenerate sites with the idea of, could it be a mutagen providing genetic changes within the population and thus higher genetic diversity, more substrate for selection. So that's the idea I'm eventually going to test with this data. I've also showed you that we do have temporal population changes in chromatin structure, so we get big changes in chromatin at the population level again over time, but not so much in DNA methylation. So there are changes between populations in DNA methylation, but it's a handful, it's very few. There's not really DNA methylation population structure. And then the exciting bit, I think, is that I've just also showed you that populations who have historical exposure in their ancestors to pesticides have a big DNA methylation response when you expose them to pesticides again, which I think is pretty cool. And then last slide. So as I mentioned at the start, I am starting my lab at the University of Leicester. I've just started. And so I'll be looking for PhD students around December time this year. So if you are interested, please do reach out. I just put a little group photo there. It doesn't really convey this, but we go on this annual retreat every year as a kind of group within the department, and it's a really, really nice working environment. So I just wanted to leave that on there because not all environments are great. This one is fantastic. So please do come and chat with me if you're interested. That's it. Thank you. Yeah.
Very cool work. Towards the end, I was curious, when you saw these methylation differences for pesticide, historical pesticide exposure,
Yep.
did that lead to any fitness differences of like survival of those things?
So there were differences, but not benefits. So the population that was actually historically exposed to pesticides in the wild is just really unfit compared to the others. It doesn't do well at all. Whether you expose it to pesticides or keep it in controlled conditions, it's not a happy population. Yeah. Yeah. Yeah.
I just wonder if the proposed methylation is mutagenesis, would you expect methylation more heavy in combined with conserved genes or more novel genes?
Great question. Yeah, I wouldn't expect it in the conserved genes really, and that is where we see it. So it's a conflicting idea. So again, we have the higher methylation in the more stably expressed genes, but then you may say if you're trying to adapt to a new environment, maybe these are the ones you need to tweak because they're core, they're on all the time, and so maybe you need to tweak them for your environment. So conceptually, I think it could go either way. Really good question. Yeah. Yeah.
Just wondering if you look at the DNA methylation in the population previously exposed to pesticide and compare that with the not being exposed to pesticide before you expose them again to pesticide.
Yes, we have. So we've got all the control data for this experiment, and there are differences. I think it's in about a couple of hundred genes, methylation differences, and a few of those genes were involved in stress response, for example. So the production of males, which is a stressful thing. So yeah, we do see differences there as well. Thank you. Okay. Thank you very much.
So a bit of a spoiler, we're going to stay on crustaceans, which is pretty exciting. I usually like to pace around when I do this. I'm going to try to convince myself not to do that so I don't walk off this little stage foolishly.
Thank you all so much for being here. My name's Zach Hancock. I'm a postdoc at the University of Michigan, and I'm very excited to be talking to you today about taking a population genetics approach to range evolution. So in 1922, the famed ecologist Joseph Grinnell envisioned species ranges as a kind of living organism, with the center he pitched as the species metropolis. This is where the vast majority of all the individuals were. This is where they were the most highly adapted to, etc. And then density kind of decreased as a function of distance towards the edge. And once you got to the edge of the range, the only thing you found is what he called accidentals. These were these random migrants that would get out to the edge, and their role was to sort of explore and expand the range if they possibly could. And so ranges become this sort of amorphous entity, kind of like an amoeba, with the tentacles sort of expanding and contracting. Now, we have kind of formalized this into what we now call the abundant center hypothesis, or sometimes the central marginal hypothesis. And it posits that the center of the range has the highest population density, and then it decreases towards the edge. So this is an empirical example of this. This is from the scissor-tail flycatcher. The region in red has the highest abundance, and then the blue is the lowest abundance. And so you can see that the population sort of has the highest in the center, and then it decreases out towards the edge. And I think that this makes a lot of intuitive sense for many of us, but the idea is not without critics. And so a lot of recent reviews have sort of questioned the ubiquity of the abundant center hypothesis. And while there are many sort of theoretical and methodological considerations, perhaps the most important and most pernicious is that ranges are often extremely complex, both spatially and temporally. So this is an example from the wood warbler, with the colors representing the different abundances in parts of the range throughout the year. And so at any point, what might count as the center or the edge of that range is a kind of moving target. It's difficult to nail down exactly where it is. And so that makes testing the abundant center hypothesis very difficult to do in practice. So to get around this, we wanted to test this hypothesis in an organism that has a very simple range in which the center and the edges are clearly identifiable and have changed minimally through time. So the organism we decided to use are beach-dwelling amphipod crustaceans. So they don't actually have a formal common name, so we've taken to calling them shovel bugs. And we call them shovel bugs because their front legs are actually modified into these like digging little structures, right? So they actually look like little shovels. They live in the swash zone of the beach. This is where the sand is not completely dry, but it's also not inundated underwater. They live on open coasts, so effectively they have a one-dimensional range, and the species that we're working on live continuously from central Florida up to about southern Maine. Okay, so our question is, what are the pattern of relatedness across this range? This first part is important to ensure that there isn't any like cryptic structure, discrete structure, and that we are in fact dealing with just a single population. Secondly is, do we find evidence for an abundant range center? And then what factors are generating the observed patterns? Okay, so what factors are, Okay, so the methods, we wanted to characterize the edges by attempting to sample both well below and well above the known boundary. So basically, I started off in southern Florida, well below where they were known to appear, and then I just beach hopped until I started finding them. And the same thing I did in southern Maine. I just continued sampling far north until long after they had already disappeared. It ended up with 15 localities and 64 individuals. We did reduced representation sequencing. We did have to remove some highly divergent individuals and populations because this family is known for having lots of cryptic diversity. And then we ended up with around 7,600 biallelic SNPs after filtering. Okay, so first, what was the pattern of relatedness that we found across that range? So we performed a principal component analysis, and it reveals both patterns of isolation by distance as well as potentially some discrete structures. So the colors here in the PCA correspond to sample site colors on the map that are colored by latitude. So if we just kind of zoom in, we see there's effectively three clusters. There's a small Florida cluster, sort of a broad smear that is the Carolinas, and then if we jump across that first PC axis, we get all the populations that are north of the Chesapeake, and I've marked the Chesapeake here on the map. So effectively, IBD is being captured in the second axis, and then potentially some discrete structure between the south and the north is being captured on that first PC axis. However, we do have a sampling gap right here in the Chesapeake, and so what we thought this might be reflecting is simply that we're just missing samples that would have connected these populations. So we wanted to know, is this discrete structure or is it a sampling gap? So to test this, we performed a triangle plot analysis, which works like this. So the hybrid index is along the x-axis, and then interclass heterozygosity is along the y. And so if you have two populations with discrete structure, as the hybrid index increases, the interclass heterozygosity must also increase. So for example, a hybrid index of 0.5 would correspond to an F1 hybrid. And so an F1 hybrid must have an interclass heterozygosity of 0.5. So it should just follow that dashed line. However, all of our populations fall well below this, and this pattern is indicative of just what you would expect if it's just isolation by distance alone. Similarly, when we plot linearized FST against geographic distance, we don't see any discrete breaks between the comparisons. So the blue and the green are comparisons within, and then the red are the comparisons between. And again, you don't see any jumps, you don't see any breaks. It looks like just a continuous increase in divergence across distance. Okay, so do we find evidence for an abundant range center now that we're pretty sure that this is just one continuous population? And indeed we do. We see that genetic diversity is the highest in the center of the range, and it decreases as a function of distance. So the x-axis here is latitude, the y is genetic diversity. So if you start in Florida, you have really low diversity. You walk your way up to the Chesapeake, diversity kind of plateaus, and then it begins to decline again as you get towards Maine. So this pattern is consistent, exactly what we would expect if the abundant center hypothesis is the best explanation for this range. Similarly, we see that inbreeding increases linearly as a function of distance from the center of the range. So x here is the distance from the center, the y is the inbreeding coefficient, and we see a strong positive relationship between the two. Once again, consistent with the abundant center. Okay, so the last thing we were interested in are what sort of factors might be generating the patterns that we observed. And to bring back Grinnell's words, what is bounding the reach of the amoeba, right? What is keeping those accidentals from expanding this population out further? And one thing that marine biologists often think about with respect to ranges and migration dynamics is what about the current regimes, right? How are the dominant currents in the Atlantic possibly shaping where migration can occur and what this range might actually look like? So in the North Atlantic, the dominant current regime is the Gulf Stream, and it runs from the loop current, which comes out from the Gulf of Mexico, kind of shoots out across Florida, follows the coastline up until about the Chesapeake, and then is diverted out into the mid-Atlantic. Similarly, there's a north to south current coming from Labrador. It's much weaker, but it follows Nova Scotia, down across New England, and then is also diverted out into the mid-Atlantic. So you basically have two current regimes, one running south to north, one running north to south, meeting in the center of the range, and then diverting out. So potentially, what we were curious about is, could the current regimes themselves be generating this abundant center pattern? Could it be that it's not actually the Grinnellian view of some species metropolis and accidentals going out, but rather the currents pushing individuals back in? So the range could be bigger if it weren't for the current regimes pushing them back towards the center. So to test this, we use Ben Peter and Monty Slotkin's directionality index, psi, which uses information from the site frequency spectrum to identify signatures of a range expansion and the directionality of that expansion. So for example, when it's just isolation by distance, it's kind of dictating the dynamics of the range, then psi should be 0. But when there's a signature of an expansion, psi increases as a function of distance from the origin of that expansion. Okay, so when we applied this to the data itself, we got some pretty interesting and weird results. So the darker colors here represent low values of psi, the lighter colors represent higher values of psi. And then I've just plotted the sampling sites. The colors here in the ocean are just the velocity so that you can see that dominant Gulf Stream running north to south. South to north, excuse me. Okay, so I'm going to interpret this for you. So basically what psi inferred was that there was two origins of expansion. So if you kind of cut this range in half with south and north. The origins of the expansion are marked in X, and then you're getting increasing values towards the center. And so what this means is that you've got more recent colonizations as you're moving towards the center of the range. And while this sort of makes sense if you think about like the dominant current regimes carrying the edge individuals constantly towards the center and this sort of asymmetric migration pattern, this struck us as pretty odd. And the reason is because often these methods for detecting range expansions rely on signatures of increased drift as you're moving away from the origin of the expansion, right? And so we were curious in how this is actually picking up on this, and what we're hypothesizing is that perhaps the effects of like strong asymmetric migration from the current regimes are actually canceling out the effects of increased drift along the edge. But this is something that we're interested in trying to test further, probably using sort of a simulation approach. Okay, so in summary and future directions, we see strong population structure characterized by just isolation by distance. We see evidence for an abundant center from both differences in genetic diversity and higher rates of inbreeding at the edge. We see this curious signal in the directionality index that appears to follow the dominant current regimes. And then we want to further test this directionality index against simulations with sort of different scenarios of directional migration to see where it will infer the origins and the more recent colonization. Okay, so I want to acknowledge my lab members as well as affiliates of the lab that gave lots of helpful feedback on this. My collaborators, Nicole Adams, Perry Wood, and Gideon Bradford. And I want to give a special shout out to Leonard Jones. He is the one that actually coined the term shovel bugs for these things. We were sitting around just trying to spitball ideas of what we could call them, and he came up with shovel bugs, and that is what has kind of stuck. And then thank my funding agency of the National Institute of Health. And with that, I'll take any questions.
You haven't talked about history. Was there a pattern of recolonization of those species after glaciation that could influence some of your results?
Potentially, but I don't know if it would be in the direction that we see, right? And that you have this both like north to south. Like maybe you would expect that to be the case from just north continuously going south if it's a pattern of like glaciation. But we see this like alternate, like two opposing patterns, which is very surprising to me. But yeah, it's definitely possible that there's some influence there. Yeah.
Is there a larval stage?
It does not have a larval stage. They're direct developers, and they're just released right into the sand. So they're not super strongly impacted by currents, yeah. Yes?
Do you have any idea about
Yeah, so qualitatively, anecdotally, they are more abundant. They're easier to collect in the middle than they are the edge. It's very hard to find them at the edge. So you do just kind of see that in the sampling. It gets easier. But I don't have like quantitative data for that. All right, thank you.
All right, great. So thank you everyone for being here with me today. So I'll be sharing some pretty relatively recent work from my PhD. So this was work done with Mike Whitlock, at the University of British Columbia. And so this project was sort of on understanding genetic differentiation in an island model under background selection. So to start, what exactly is background selection? So background selection is the process by which selection on deleterious alleles also affects linked non-deleterious alleles. And so this is illustrated in this cartoon below. So on the very left, you can imagine we have some cartoon haplotypes. Mutations that occur in the yellow region will have deleterious effects on fitness, so these are marked in red. Mutations that occur in the surrounding region will have neutral or no effects on fitness. And so we expect that these mutations of deleterious effects, so individuals with these mutations will eventually be selected out of the population just because they're less fit. Now, consequently, if these blue neutral mutations marked in X's in the middle, if these are associated with or linked to these deleterious mutations, we also expect them to be removed from the population and feel this action of linked selection. Hence, background selection tends to reduce genetic diversity. And this is typically why we not only see reduced genetic diversity at genic sites, we also tend to observe reduced genetic diversity surrounding our genic sites because of these neutral alleles being dragged down by this effective background selection. So we typically measure this effect of background selection using this metric B, which can be defined a couple ways. So it can be defined as your ratio of your effective population size, so with background selection, to your census population size. So this sort of captures the idea that selection against deleterious alleles indirectly acting on these neutral alleles accelerates the rate of drift at these neutral sites, and so it reduces the effective population size. So with more restrictive assumptions, you can then use this to estimate genetic diversity pi, so it can also be defined as pi over pi naught, which is your neutral diversity with background selection divided by your neutral diversity at a locus without background selection. So, well, why do we really care about background selection? Well, when we consider the selective effects of most mutations, we know that they're mostly deleterious as opposed to beneficial. And so it follows that background selection is essentially a ubiquitous process affecting patterns of genetic diversity among pretty much all living species. So here is a map of B, so genetic pi over pi naught in the Drosophila melanogaster genome. Don't worry too much about the lines and the axes. This is sort of just to illustrate that there's a lot of variation in B that you can observe all across essentially the entire genome. It's also been measured in humans, so this is chromosome 2 in humans, and that's the wrong reference on the bottom right. This is Buffalo and Kern 2024. But you can see that there's also widespread variation in B across human chromosomes. It's been estimated in a lot of other species. So it's a widespread thing that we do know about. So as I just said, background selection tends to vary quite a bit across the genome. And this is consequential because what that means is background selection may be sort of a confounding process, confounding tests of other genomic tests looking for other processes, such as positive selection, locally adaptive alleles, or even trying to estimate demographic parameters. So it's something we kind of want to understand. Now here's sort of the caveat. So the background selection models that have been used to make predictions in empirical data mostly assume panmictic or randomly mating populations or really low rates of migration. But all of us as biologists in the room know that most natural populations exhibit some form of non-trivial population structure. And so this is something we kind of want to explore more. So in particular, what Mike and I were interested in was, well, how does background selection affect rights fixation index FST? So FST is a commonly used measure of genetic differentiation. It can be defined a multitude of ways. So you can define it as the standardized genetic variance among subpopulations, or 1 minus the ratio of subpopulation and total population diversity. So an FST of 1 meaning that your populations are completely genetically different, and an FST of 0 meaning they're panmictic. And so FST is really useful because you can use it to estimate both demographic and genetic properties of a metapopulation. So demographically, you can use it to assess the degree of population structure, the time since subpopulations have split from some ancestral population, or genetically, you can use it to infer genetic differentiation and hybridization rates, and even spatially varying forms of selection. So first, we sort of turn to the literature to understand, well, what do we know about how background selection affects FST? And so there has been some work done in an island model, so that's illustrated in the middle. So an island model consists of some number of demes of equal and constant population size, equal and constant migration rates, non-overlapping generations. And so on the left, you recover the classic result for FST at equilibrium without background selection. And we can see that it just depends on your local population size and your migration rate. If you increase your migration rate or population size, that decreases FST. And you can think of that in this model as just an increased gene flow. So Zheng and Corcoran showed that with background selection, we can actually approximate the effects of background selection on FST by modifying our local population size by this factor B. And so essentially what we're doing, going back to that previous slide, is we're recovering the effective population size. And so we kind of know that migration affects FST, and background selection can modify that factor, but we don't know if migration affects B itself. And so that sort of brings us to the sort of central question of this talk, which is, well, does migration influence the effect of background selection on FST and not just FST itself? So to ask this question, we first turned to some simulations. So we ran simulations in SLIM. We simulated a relatively simple genome. So we have a neutral site embedded in a region subject to deleterious mutation. So mutations occurred at some fixed rate with some fixed heterozygous selection coefficient. So these were in diploids. And the region had some total recombination rate. And we were interested in what is B at that neutral locus in the middle and what's FST. So here's what those simulations look like. So just to orient you, on the y-axis is FST, on the x-axis is our heterozygous strength of selection scaled by our local population size. Each of the gray dots represent mean FST from replicate simulations. So on the left, in our very low migration rate, we sort of recover this inverted U-shaped curve of background selection with respect to varying strengths of selection. So we see a peak in background selection at some intermediate strength of selection. And this is sort of in line with what we expect from the theory. I don't have time to talk about that as much, but it's what we expect from the Zeng and Corcoran predictions. On the right, on the other hand, we have our very high migration rate case. And here we actually don't recover that inverted U-shaped curve. Instead, we just see a flat line of FST with respect to varying strengths of selection. So sort of suggesting that there's no effect of background selection on FST for any strength of selection under our very high migration rate case. And so we recovered some appreciable evidence that migration does influence the effect of background selection on FST. And so our next question is, well, can we derive novel theory to account for this effect? And so the way we sort of thought about this was, well, going back to our original slide, we consider that the effects of background selection on our neutral allele depends on the degree of association between that neutral allele and the deleterious allele. If they're not that associated with one another, we don't expect much effective linked selection. Conversely, if they're tightly linked, their fates are essentially intertwined. And so we're going to do a thought exercise. We're going to use a charismatic organism, the rectangle. And so it's a diploid organism. We have a neutral and deleterious allele on the same background, so they're associated with one another. And we're going to consider what factors would potentially break up this association or end the association between our neutral and deleterious allele within within a subpopulation within a metapopulation. So we consider one lineage. That lineage has some offspring. They have their own offspring. And then eventually they fail to reproduce, and that's just the action of natural selection. So once that haplotype gets removed from the population, that association has now ended between our neutral and deleterious allele. And this we can just track by knowing the selection coefficient, T. Next, we consider another lineage that has some offspring, and those offspring have their own offspring. And if some recombination event happens in the gametes that are passed down, now it's your deleterious and neutral allele will be passed on separately. And so you've lessened or ended the association between your neutral and deleterious allele. And this we can track by just knowing the recombination rate between our neutral and deleterious loci. So thus far, all of this has been well characterized by the background selection literature. We kind of know what's going on here. So here I'm going to add the additional expectation that if we have another lineage and that lineage has some offspring, those offspring have their own offspring, and they simply migrate out of the population into another, or migrate out of the subpopulation into another subpopulation, that ends the association of that neutral and deleterious allele within the subpopulation we're looking at. So it still exists elsewhere, but within our focal subpopulation, that association has ended. And this we can track by just knowing the migration rate in our island model. And so we did some math. Voila. We go from the left equation to predict B to the right formula to predict B, including the effects of migration. And then our next question was, well, how well does this predict what we saw in our simulations? So here are the simulations I showed you all a bit earlier. So the gray dots being the observed result from the simulations, and now the pink dots overlaid are our predictions from our theory. And it does quite well, so we're quite confident that this is what is happening in an island model. So just to conclude what I talked about, so hopefully I convinced you all that migration reduces the effect of background selection on genetic differentiation, and we have derived novel theory to account for this, so we can predict it quite well. One sort of empirical prediction of this work is that when migration rates are sufficiently high, we don't expect background selection to be a strong confounding factor for locus-to-locus FST scans. A caveat to this being that this is work done in an island model, so we're not exactly sure how it translates to more complex demographic scenarios. So with that, I'd like to just thank and acknowledge members of the Whitlock Lab and adjacent NSERC for the funding. We do have a preprint up. Substantive revisions are underway and due on Wednesday. I did have a QR code in the bottom right, but my free trial expired, so I'm sorry. But yeah, thank you. Any questions? Yeah.
So very nice talk.
Thank you.
This is the classic background selection model, right?
No, so we incorporate weak selection into the model.
Yeah, but it's like it's built on the foundation of the Charles
Yeah.
model, right?
Yeah.
So that one assumes that the selection is quite moderate, not weak, right?
Yeah, so this is a modification of that theory. So we use a more recent framework. So this was developed by Santiago and Caballero. And what this one does is it sort of tries to account for weak selection by considering that weakly deleterious alleles just fix at some rate, and they no longer contribute fitness variance. And so we can kind of account for that. So
Yeah, because it looked nice also in the low.
yeah, if I put the Charlesworth one, it goes like to the sky. Yeah, when you go to lower strengths of selection. Okay, thank you.
Yeah, please.